I will introduce myself. So thank you to those of you who are sticking around for the final session. It's almost everyone, which is fantastic. It's been a great meeting so far. My name's Lana. I'm an ID physician and microbiologist at the PA, and it's my pleasure to kick off the section on emerging diagnostic tests. We've got some great upcoming speakers, and I've just realised that the year 2014 is an auspicious year for all of these people. So firstly, I'd like to introduce Dr. Mariana Pitombeira Liborio, who is a Brazilian ID physician who is currently in Brisbane completing her PhD looking at metagenomics in children with sepsis. Mariana first fell in love with Brisbane in 2014 and held onto the desire to return and came back here in 2024. We are privileged to have her speaking today. Thanks, Mariana.
Thanks for having me here. So I'm going to talk a bit about metagenomics for bloodstream infection diagnostics. So first of all, the importance of bloodstream infections. So we cannot see that, but the global burden of bloodstream infections. It's a major cause of morbidity and mortality in the whole world. It's common, it's life-threatening, and it's increasing. And we have an estimated 150 cases per 100,000 population and a crude mortality of 17% for bloodstream infections. It's also a major contributor to sepsis, which we have in 2021, we had 166 million cases and 21.4 million deaths, which accounts for 31.5% of all global deaths. And it's a problem worldwide, so sepsis mortality remains high across all regions of the globe. The greatest burden of it comes with gram-negatives, but we have other pathogens such as staph and also fungi causing bloodstream infections. And another issue that comes with bloodstream infections is the increase in antimicrobial resistance that was already cited here quite often. But there is an interesting document from the World Health Organization that was released last year, analyzing the period from 2018 to 2023. And we had in this period an annual increase of 5 to 15% in antimicrobial resistance. So it's quite daunting. And why does bloodstream infection matter? So it's, as I said, it's a problem in the whole world, but its mortality is especially high in specific populations. So older adults, infants and neonates, immunosuppressed, which are transplant patients, chronic comorbidities, and also in lower and middle income countries, we find higher mortality rates. And recently, we had a systematic review. One of our colleagues, Anna Perry, did a systematic review where she compared rapid diagnostic tests for bloodstream infections. And she found that if you use these tests and pair them with stewardship antimicrobial programs, you reduce time to optimal therapy, and this can improve survival in these patients. So specifically in the transplant patient, if you think about the stem cell transplant patients, you can have an overall incidence of BSI of up to 35%, 100 days in the first 100 days after the transplant. And if this bacteremia is by a gram-negative multi-drug resistance, you can have up to 60% 12-month mortality. If you take a look at cohorts of solid organ transplant mortalities, you see that the BSI mortality for multi-drug resistance organism varies a lot from 15 to 82%, but it's always consistently higher than with non-resistant organisms. And the factors that are in these patients that cause this increased susceptibility are immunosuppression, as was already talked about here a lot, mucosal barrier injury, facilitated translocation, central line and other catheters facilitating invasion, and also the rise of this MDR prevalence. And what we currently use for diagnosis bloodstream infections, as we all know, is the blood culture. It's the standard, it's well established, it's been used for years. We have established protocols and interpretation guidelines. It's inexpensive if you think about the whole apparatus that we have in the lab. but it also has poor sensitivity, especially if the patient has already used antibiotics, and it can be time consuming. So it can be as fast as a few hours, but it can take up to 3 days for you to have the results. And we also have currently rapid targeted panels that are also used currently, and they shorten time to identification, but they have this restriction of being panel limited. So you search for specific genes, and if the organism or the resistant gene is not in the panel, it will not be detected. So this is what we currently have. And so this might take a long time. So first we have the blood culture collected. If it comes positive, we do the gram stain, growth agar, identification, antimicrobial susceptibility, and other testings. But sometimes it doesn't even become positive, even if we know that there is something there, and we need to find out what's in there. And so there is next generation sequencing. Next generation sequencing is when you sequence, you analyze thousands to millions of DNA fragments from a sample in a single run. It has some specific characteristics. So you can use it for pathogen detection, including polymicrobial infections. You can identify resistance genes. It's considered a rapid analysis. It can identify pathogens that are missed by culture or organisms that don't grow in the culture. It's less affected by antibiotic exposure, although it's still a bit affected. And in a single test, you can detect bacteria, viruses, fungi, and protozoa. And this is a bit of the workflow, the basic, it kind of changes a lot. It can be tweaked a lot, but the basic workflow is that you have the clinical specimen. You extract the nucleic acid from that. You prepare the library, you sequence this library, and there are two main platforms for sequencing, which is Illumina and Nanopore, so short reads and long reads. Then there's data analysis, bioinformatics, and then the report to the physician. And there are three main types of next generation sequencing. So one is whole genome sequencing, which still relies on culture. So you need to culture the organism and then study the whole genome and see if it matches a database that we have and identify the organism. There is targeted next generation sequencing, which you do from a sample, but then it's as those targeted panels, you target specific genes. You try to find specific genes in that sample that would help you identify if there is a specific pathogen there. And there is metagenomics. So metagenomic, you get a sample. It can be cerebrospinal fluid, like in this example, but it can be blood. It can be plasma, can be lung sputum, can be pretty much any sample, any bodily sample. And then you sequence all nucleic acids containing this sample. So human, bacterial, virals, everything. And using bioinformatics, you identify which one is which. So I have DNA, for example, for human and this specific bacteria here. And then I can say that this patient has an infection with this specific bacteria. So these are the main types. I'm going to focus more on metagenomics. So remember, sequencing all the nucleic acids. So it's important to understand the next steps. So this is the performance of the metagenomics. So detection, it detects DNA. So detection is frequently higher than culture. And usually in the studies that have been done, it's been able to change therapy. So you get those results, you compare that to the clinical scenario of the patient, and okay, let's broaden, let's change the antimicrobial treatment, or let's stop antimicrobial treatment. That's been done already. So I brought two examples. So this one was a retrospective study with hematological patients. So I had more than 300 patients, neutropenic and non-neutropenic. And the overall positive detection rate of plasma cell-free DNA was significantly higher than of conventional microbiology tests. And I brought this on purpose because there is this possibility of doing the sequencing of plasma. There is, when you have an infection in any time, in any part of the body, the organisms, they release small pieces of DNA, and this can be measured in the plasma. So it's plasma cell-free DNA. It's sometimes called liquid biopsy because you can identify an infection in any part of the body just looking at what DNA is in the plasma. And another interesting thing is that it's really important to have a clinical panel to adjudicate the results of these tests, because if you're comparing to a standard that's not so sensitive, and then you find a higher positive rate, is that higher positive rate real or not? So it's really important to bear that in mind. This second one was a prospective study with leukemia patients and febrile neutropenia patients. And they compared standard microbiological tests with the Carios test, which is another, it's a commercial type of cell-free DNA test. And it was twice as likely to provide a microbiological diagnosis, often without resorting to invasive diagnostic procedures. So that's this advantage of through plasma, through a blood draw, you can say whether there is an infection in the lung or in this nervous system, any part of the body. So specifically in transplant and immunocompromised hosts, there are some papers and some research being done. So this first one it grouped transplant and hematology patients and patients who went through stem cell transplant. It grouped plasmas, cell-free DNA, and also metagenomics from a sample where you usually study genomic DNA, so the DNA that's in the chromosome, not the plasma's pieces of DNA. And depending on whether you use plasma cell-free DNA or genomic DNA, and also depending on what's the febrile syndrome that the patient has, you can have results detection ranging from 25 to 60%. And in lung transplant recipient, it was done using sputum collected by bronchoscopy, and it could shorten turnaround time compared to culture. And also it was able to change the management, either to run more specific tests or to change the antimicrobial treatment in almost 30% of the cohort. This was another study with a pediatric stem cell transplant recipients. They used cell-free DNA to detect when they had febrile episodes. And the positivity of the metagenomics next generation sequencing was 58%, while the culture was 13%. So in this specific study, these results of the metagenomics helped adjust the therapy in half of the patients, either stopping or starting or antibiotics or ordering exams or avoiding exams as well. And this was the maximum turnaround time. It was 31 for metagenomics, and it's up to 70 hours for culture-based methods. And another study that was quite interesting that I thought it was interesting to bring is they did a study with patients with leukemia and patients from stem cell transplant recipients, and they had bloodstream infections. And they looked back in the plasma of these patients, and they saw how early they could detect the DNA of that pathogen that caused BSI. And they could detect the DNA as early as 2 days before the onset of the infection. So it kind of brings possibilities of what we can do with these tests. So with this, the group here at UQCCR previously did the DETECT study. So where they got positive blood cultures and tested different extraction methods to extract the DNA. There is a very important challenge when you're dealing with blood and blood culture is the amount of host DNA compared to bacterial or pathogen DNA. So when you have a bloodstream infection, usually you have less than 50 CFU per mil of bacteria, for example. So if you think about metagenomics, where you see the whole DNA that's there, the bulk of DNA will be human. And there will be just a little bit of bacterial DNA, and it will be kind of hard to find. But it's there, or you believe it's there. And so many different extraction methods were tested and different platforms. So rapid multiplex PCR tests, nanopore, which kind of sequencing of next generation sequencing, and Illumina sequencing. And these results, so the nanopore and the Illumina results, they have to go through an bioinformatic pipeline to give us the results. And this was done through a locally developed platform called RS Cloud. And in this DETECT study, using a specific DNA extraction method where you do the host depletion and then extract the bacterial DNA and running it through the RS Cloud, it could identify a species in 38 of 40 samples. So it matched the result of the positive blood culture in 38 cases. These 2 cases that didn't match were 2 that were polymicrobial, and so they were missed by the second pathogen was missed by next generation sequencing, unfortunately, but it was promising. And then another step was done. So this was one with that included adults and children from the intensive care units, and also used their positive blood culture and compared the conventional workflow to identify pathogen and AMR resistance with a workflow with Nanopore, which is another platform for sequencing. So we used the same DNA extraction method that had worked in the previous work. And instead of using Illumina, used Nanopore. So it has faster turnaround time versus Illumina, so you can have results in real time. So it's when you want faster results, it's a nice thing to try. And also, again, it had 100% concordance with species ID if it was monomicrobial. But for antimicrobial resistance, it was not as good as we would expect. So it had up to 12% of very major error. But for some specific species like Staphylococcus aureus, it had a categorical agreement of more than 90%. And it's interesting that these results from these studies, they were available within 8 to 16 hours after blood culture positivity. So it's promising, it's encouraging. And then with these comes to my PhD study, which is the DECA study, which we want to accelerate pathogen identification and antimicrobial resistance prediction using next generation sequencing. So at the QCH, the Children's Hospital, we are following children who are under the care of oncology. So both children with cancer and children who undergo stem cell transplantation. And whenever they have an episode of sepsis or suspected sepsis and have bloods collected for blood cultures, we collect bloods for next generation sequencing tests. And we want to compare these results and also how fast this can be done. So we are using next generation sequencing tests for both in positive blood culture, as it has been done before in those previous studies, but also using for direct from blood. So we are trying to see if we can use these methods to not have to wait until a positive blood culture and see if we can detect directly from blood. And it's very challenging because, as I said previously, the amount of DNA compared to the human DNA is very little. And there's also be a part with host response study, but that is not part of my PhD. I won't talk much about that. And part of this DECA study is the development of the methods in the lab as well. So we saw that it's possible to do it to identify using metagenomics, identify bloodstream infection using metagenomics from positive blood culture. We are trying to do it from whole blood, which is quite difficult. And then what if we do something in the middle? What if we put the culture to incubate, and before it flags positive, I take a sample and see if I can identify any pathogen there. So what we've done, we got an E. coli with the resistance gene CTX-M15. We put less than 50 CFU per mil in blood and in the blood culture bottle. As I said, this is usually the amount of bacteria that you get when you have sepsis directly from the blood. And we incubate and take samples at different times. And then we do host DNA depletion, bacterial DNA extraction, run them on the Merino. It's a flow cell from that uses nanopores for sequencing, and run their own bioinformatic platform that's Epi2Me, and then see how fast we can identify bacteria or AMR genes. So in this specific case, we know what we have. So we know that we put this, and I can see how early I can find this here in this bloodstream infection that I prepared. And this is what the Epi2Me system gives to us after the 72 hours of sequencing. So it can go up to 72 hours of sequencing. So these are the samples. So each bar here is a sample. And so this one is one collected after 1 hour, 2 hours, 3 hours of incubation. So I let it incubate for 1 hour, I let it incubate for 2 hours, 3 hours. This side of the panel is the ones that I used the method previously cited to extract bacterial DNA, so host depletion in bacterial DNA. This one is another method that we saw that would be promising as well. So also for host depletion in bacterial DNA extraction, so basic 5 in Biostic. This one is basic 5 in DNeasy. And the blue color is the amount of human reads in the sample. The orange is the amount of E. coli reads in the sample, and the green is others. So it's contaminants. And another important thing is these contaminants can be anywhere. So they can be in the kit itself, because it's DNA. We're detecting DNA. So it can be pretty in the kit that we use for extraction, in the kit that we use for sequencing. So it's really important to have these control here. There are the controls for the test. So they have all that we used, but not the bacteria that we added. And it's quite interesting to know that after 72 hours of sequencing, even doing host DNA depletion, you can see blue, so human, in almost all samples. So it doesn't deplete everything. So it's really hard to get rid of that to actually see the bacterial DNA. And what's really interesting about this experiment is that when we got at the sample at 5 hours and extracted using this kit, so basic 5 in Biostic, we were able to identify that there was an E. coli there when it reached 10 to the 4 CFU per mil, and only after 5 minutes of sequencing. So that means that if this was a real patient, after 5 hours and 5 minutes, in the best case scenario, because there is transport, there is all the other things, you would be able that there is an E. coli there. And after 7 hours of incubation, and then we needed more hours of sequencing, we needed 3 hours of sequencing, we could say that there was CTX-M15 gene there. So it would be 7-3, so 10 hours after sample collection, I would be able to say, yes, there is a pathogen there. Yes, there is a pathogen that's probably an ESBL producer. Let's use antibiotics accordingly. It's very promising. We're working on improving that, refining that. So the practical challenges, as I said, the host DNA is overwhelming in the amount of DNA that we get, especially as we are trying to work with whole blood and incubating the whole blood. So the host depletion methods, they can kind of boost these microbial reads, but then they lower the total DNA concentration, and the nanopore that we are using is not enjoying that much. So we are trying to find the right number of the right concentration that would be optimal for that. And the interpretation of this is still very challenging. So we detect DNA. What does this detection mean? Does it mean contamination? Does it mean a commensal that's there? Is it a true pathogen? So there are some thresholds that are used, so you can kind of tell by the number of reads. The more reads you have, kind of the more true that pathogen is truly, that organism is truly a pathogen, but there is no number defined yet. And also, we also need to remember that genotype is different from phenotype. So if it has the gene, doesn't mean it is resistant. So you have to be very careful with that. So this picture is very common when you talk about metagenomics. It was published in 2019. And most of the hurdles that are there, we still have now. So there is the limitations for use are the frequent detection of colonizer and contaminants, or if we detect, are they colonizer, are they contaminants? We still don't have a threshold to define that. The high human nucleic acid background of blood. Appropriate timing of testing. Am I testing 2 days before? Am I testing preemptively? Am I testing when the symptoms arrive? Is it best to test 2 days later? We don't know yet. So we don't have reference standards. There is a need for bioinformatics expertise. So the system, it only gives you general information. If you want to pinpoint at which time you can find that pathogen specific, like 5-minute sequencing, 3 hours of sequencing, you need to have a bioinformatician by your side. The dedicated facilities and infrastructure, and it's still very expensive. So it's not for every patient. You would have to have selected patients to choose to use that. So in conclusion, so the BSI remains a major driver of mortality, global sepsis mortality, and we still have to think about antimicrobial resistance. So metagenomics can improve detection and time to answer, including in culture negative, pretreated, and transplant patients when applied judiciously. So not every patient. And there are many things that we have to handle to be successful. So host DNA management, contamination control, interpretation, and also diagnostic stewardship to really understand what these results mean. Thank you.